Now let's move towards the very final stages of TCA cycle. The succinate which was produced in the previous reaction will now be oxidized, and it will be oxidized to give rise to the formation of fumarate. The enzyme which is used here is succinate dehydrogenase, and also importantly, from this oxidation reaction we get FADH2 from FAD. Also, you should keep in mind that succinate dehydrogenase is the only enzyme which is embedded into the mitochondrial membrane. If you remember our discussion on the electron transport chain, you remember that the succinate dehydrogenase complex makes the complex number 2 of the TCA. There are 4 complexes of TCA. This complex number 2 is formed, and it is involved in the donation and carrying of the electrons. Then fumarate is hydrated to give rise to the formation of malate. The enzyme here is what we say the fumarase enzyme, and the water is added here. That's why we say it's the hydration process. In addition to succinate, fumarate is also produced by urea cycle during purine synthesis, and also through the catabolism of phenylalanine and tyrosine. Then in the very final reaction of TCA, malate is oxidized into oxaloacetate. The enzyme here is malate dehydrogenase, and again NADH2 is formed here, which is the third and final time that NADH2 will be formed from TCA. Also keep in mind something very interesting: the standard free energy of this reaction is positive. Then how does this reaction occur? Actually, this reaction occurs because of the very highly negative standard free energy of the citrate, of the formation of the citrate through citrate synthase. And if you remember one of our previous topics on the Gibbs free energy, I told you one of the very important feature of Gibbs free energy, or the standard Gibbs free energy, is that it can be added up. I mean, it is additive in nature. So if there are 7 or 8 different reactions you are talking about, the reactions, some of the reaction may have a positive delta G value, that they will be non-spontaneous. Some may have the negative delta G value. If all of the delta G value is negative, the reaction will occur. So this is how the negative delta G value of overall reactions make the reaction possible. Oxaloacetate or oxaloacetic acid is also produced by the transamination of amino acid aspartic acid. So now you are seeing here that not only these intermediates are formed during different reactions of the TCA, but they are also being formed from other pathways, and they then become part of the TCA cycle. We will elaborate on this phenomena a little bit in more detail later. कुछ सवाल जो हमें पूछने चाहिए, जो पूछने बनते हैं, आए सोचें. Why? It's very easy one, by the way. Why TCA have to takes place in mitochondria? Why not in cytoplasm? रिलेशनशिप क्या है? बार बार हम इलेक्ट्रॉन ट्रांसपोर्ट चेन की तरफ जा रहे हैं, ग्लाइकोलिसिस में भी, TCA में भी, और TCA और स्पेसिफिकली TCA और ETC, इलेक्ट्रॉन ट्रांसपोर्ट चेन, इनमें ताल्लुक आपस में है क्या? What is the major factor which make TCA so highly energetic? I mean, so highly energy giving metabolic pathway. What is the major factor? It looks simple, but I really want you to ponder on it, that why we say TCA as cycle, tricarboxylic acid cycle. इस वर्ड का क्या अहमियत है साइकिल की? Why we say it cycle?